We are the Plant Antimicrobial Discovery Team. Our team brings together a diverse array of expertise and backgrounds. Still, we are interested in the broad topic of antimicrobial resistance. The overarching question of our project is, can we create an affordable, multi-part kit setup to test the antimicrobial capacity of plants and plant extracts? Disease-causing microbes are evolving new resistant mechanisms that challenge the efficacy of currently available antimicrobials. These resistant microbes spread globally, causing diseases such as tuberculosis and gonorrhea that do not respond to current treatments. Antimicrobial resistance happens when microorganisms like bacteria, viruses, and fungi change over time and no longer respond to existing medicine. Antimicrobial resistance makes it harder to treat, reduce the disease spread, and cause severe illness and death. According to the United Nations, it is expected that antimicrobial resistant diseases could generate up to 10 million human deaths each year by 2050. Just like the large number of microbes that cause disease, the causes of antimicrobial resistance are multifactorial. They include excessive usage of antimicrobials, lack of adequate policy, and a lack of discovery of new antimicrobials. There is a paradox between the need and discovery of new antimicrobials, as it's shown by the declining interest in discovering and introducing new antimicrobial agents by the world's major pharmaceutical companies. One source for antimicrobials is plant and plant extracts, which many have shown antimicrobial properties in vitro. With over 391,000 vascular plants on Earth and an extensive traditional knowledge that spans many cultures globally, we asked ourselves if there was an affordable and mobile way to test local plants' antimicrobial capacity anywhere in the world. To accomplish this goal, we gather a group of diverse expertise to build a long-term community, which is made of scientists, educators, and public residents in different countries worldwide. Our aim is to utilize what we have called the Plant Antimicrobial Discovery Kit, so we can survey the plant's antimicrobial capacities anywhere in the world. Our project is divided into different sub-goals. One sub-goal is related to the resources that will be used to grow bacteria. We are testing different microbial growing devices and mediums, as well as different sources for non-pathogenic bacteria. Since our focus is to identify plants with antimicrobial capacity, we are also designing data collection tools that can help us gather information, such as the name, pictures, and geolocations. This presents the opportunity where data can be crowdsourced and displayed publicly for people and researchers. Finally, we would like to use this kit as an educational tool to present the concept of antimicrobial resistance to different audiences. This is a multi-part project, and it will take time for all the parts to come together.
Currently, we are focusing on developing an affordable mobile microbiological incubator. A microbiological incubator is an enclosed chamber where temperature, humidity, and air are controlled to promote microbial growth. To test if plant extracts affect bacterial growth, we have to grow microbes in a vessel and nutrient media at a warm temperature. Because the weather changes as you move anywhere globally, we thought that developing an incubator could assist in growing bacteria in the shortest amount possible without contamination issues. Incubators are a standard tool used in research laboratories. In general, they are used to maintain and propagate cell lines and microbial cultures. Many considerations are taken into account to determine which incubator is needed. Example of features are the chamber size, atmospheric control such as gases and temperature, air exchange, humidity, heating control, decontamination easiness, and monitoring capacity. Incubators vary in price depending on their specification. Basic small incubators can start in the low $500 and complex ones can go as high as USD 20,000. These prices ranges are not very permissive to many budgets, so they are often shared between research teams and facilities. In educational settings and remote areas, incubators may not be readily available and are not cost permissive. Therefore, we tasked ourselves to determine if we could prototype an affordable and portable microbial incubator. Our initial solution has risen in the form of a 3D printed microbial incubator that anyone with a 3D printing blueprints and a 3D printer can access, with an estimated cost between $47 to 75 USD. Since it is a prototype, the prices are still initially high. We expect this incubator to regulate temperature and carbon dioxide levels to maintain an internal atmosphere ideal for microbial control. The mobile incubator is about 23 centimeters tall and should weigh less than 3 pounds empty. It can hold up to 5 plastic tubes, 5 microtubes, and 3 petri dishes simultaneously. However, the holding racks can be modified to whatever the needs of the experiments are. Our incubator size also makes it portable and easily accessible to anyone, making it an economical and innovative solution to the expensive, not always available incubators. The electronic components that will control and regulate the temperature are the most considerable expense in the prototype. The heat source will be LED lights and an extractor fan to circulate the air and allow the exchange through the HEPA filtered covered vent in the incubator. Sensors will feed into an Arduino that will display information to a digital screen. We expect that this incubator will be able to self regulate its temperature. This is an initial prototype. We are still working with the scientists' feedback in our team and the electricians and engineers in the Mexico Makerspace team. This is not a final model, and we are open to continuous improvement as well as redesign. We are also working on a DIY incubator model with materials found at home using light bulbs and shoe boxes. We hope to create at least two versions that can be adapted to any setting and provide options to the volunteers that will help us gather plant antimicrobial capacity data.
We would like to thank the current team, which has been pivotal in refining the incubator prototypes. The prototype that we presented today was designed by Hugo Atla and Rafael Perez from Mexico, with contributions from me, Liz Albertorio, Deepti Shiri, Sana Jalili, Ukbat Farah, Rocio Gonzalez, and Harry Aklago. We are going to continue developing the incubator prototype long after Frugal Science class ends, and with all the other stuff that we have planned, we definitely are going to continue working on all those ideas. The Plant Antimicrobial Discovery Project was initially conceived by me, but it was a collaboration with Dr. Slavko Komanersky at North Carolina State University and his mobile discovery kit. You can find more information about our project by visiting our Notion page here on the last slide, or you can send an email to me at lizalbertorio@gmail.com if you want more information, if you have any questions or any comments, or if you want to join a team. We want to thank Manu and Tyler for this amazing opportunity. Um, I personally have made friends and found collaborators that have helped me bring forward these ideas that I had in my head, and they had made it possible. So I thank all my members of my team for their trust. And I hope that we continue long after this class ends, and we're working on that. Thank you to the other teams as well, because their ideas have also helped us and inspired us well, to what we're doing. And congratulations and good luck. And I look forward to see all those projects and ideas that you have brought forward. And it definitely has given me perspective on what frugal science can do for the world. Thank you.